Good morning. On behalf of the ALS Association Greater New York Chapter's Board, and as a member of the Chapter's Care Services Committee, it is my pleasure to welcome you to the second annual Education and Resource Summit, Navigating the ALS Journey. I joined the board of the ALS Association Greater New York Chapter in 2011, several years after my mother lost her battle with ALS at the young age of 62. Most of the members of our board have a personal connection to this disease and feel a deep sense of purpose and commitment to carry the torch forward in our efforts to raise funds for research projects, patient education, and providing resources for patients and families living with ALS. The ALS Association is the largest private funder of research worldwide. Over the past several years, we have seen significant progress due to the funding boost from the Ice Bucket Challenge, which enabled the ALS Association to fund a number of strategic research initiatives. One important initiative is right here in New York at the Genome Center, and our chapter is proud to be a partner in establishing the ALS program at the New York Genome Center. Over the next 2 days, you'll have the opportunity to take part in numerous sessions and conversations that span a wide variety of topics related to ALS. Everything from research and clinical trials to managing nutrition and very much more. No matter what is happening in the world, our mission to do whatever it takes to support the ALS community remains constant and vigilant. We at the Greater New York Chapter, board and staff are here for you to support you as we navigate this journey together. Thank you again for being here, and I hope to see you soon.
Hi, everyone. My name is Debbie Schlossberg, and I'm the Chief Care Services Officer here at the Greater New York Chapter of the ALS Association. On behalf of the Care Services team and our entire organization here in New York, I want to thank you for joining us for our second annual Education and Resource Summit. Before introducing our keynote speaker, I just have one or two housekeeping points. The first is that Dr. Harms will be sharing his presentation with us, and that will be followed by a question and answer session. So if you have questions that you'd like to pose to Dr. Harms, please use the Q&A function that you see at the bottom of your screen. I will be checking those questions for the duration of the presentation and will be asking them at the end. Also, please note that this is being recorded, as are all the sessions in our presentation, in our summit, and they will be available for future viewing. And now to introduce our keynote speaker. Dr. Matthew Harms has an extensive resume, so I'm just going to share some of the highlights. He is the Associate Professor of Neurology at Columbia University Medical Center. He's a clinician researcher whose past work has contributed to early understandings of the ALSC9-ORF gene, and he helped lead the first large-scale exome sequencing project in ALS. His current research focuses on gene identification, and he's the director of the Precision Medicine Initiative at Columbia. Dr. Harms is also chair of the ALS Expert Review Board panel for ClinGen. And now I would like to have Dr. Harms start his presentation. Welcome.
Thank you, Debbie. That was a very kind introduction. Is everyone able to see my title slide and able to hear me?
Yes.
Great. Well, it is an honor to be able to present to the patient community today. And I do look forward to a day when we're able to join with one another in the same location, potentially down at the New York Genome Center like we did pre-COVID. If we were going to do that, I would be arriving by subway, but if not, I would have to take my clunky old Honda Odyssey minivan. I would, of course, rather be showing up in something sleek and cool like this Tesla, which, of course, many of you know bears almost no resemblance to my very uncool Dadmobile. This, of course, is an electric self-driving vehicle, which has only very recently been feasible. And, you know, my van and the Tesla share a couple of things. They have very similar car bodies made out of the same materials. The tires aren't all that different. The seats are pretty similar, and they both have a radio. But in order to go from the place where you could have a minivan to the place where you can have a self-driving electric vehicle required extensive research and development. And it required that development across multiple fronts and in parallel. You needed an engine that was super highly efficient and very fast. You needed batteries that could store enough charge to go 500 miles. You needed charging stations across the country. Of course, to make it self-driving, you needed to have amazing GPS with great resolution no matter where you went in the world. High resolution cameras that could detect things ahead of you, behind you, under you, and above you. Extraordinarily fast computer processors that could take all this information in and make split-second decisions based on artificial intelligence. And then, of course, you needed hours and hours and hours to generate data, lots and lots of data to be able to figure out how to allow a self-driving vehicle. I borrowed a friend of mine's and actually drove it through the city streets of New York on the self-driving module. It was a little bit scary, but it worked, and it was absolutely unbelievable. But of course, if any one of these 7 or 8 technologies was not developing apace with the others, we wouldn't have it. We could have all the great resolution cameras, the processors, the computer, but if we didn't have GPS to localize all of that stuff, we wouldn't have self-driving cars. Without the batteries, we wouldn't have the electrical components. Without the data, we wouldn't be able to do this at all. And so really what I would like to emphasize is that great accomplishments, like a self-driving vehicle, require extensive research and development across multiple fronts in parallel, all coming to fruition. Now let's say that we want to get rid of this Tesla and replace it with what we're all here to do today, which is to develop something just as complicated and just as and even more important, effective therapies for ALS. What are the technologies and things we need to come up with in order to make this a reality? Well, we need extensive understanding of ALS, the mechanisms, the genes, the environmental exposures that lead to ALS. We need advances in medicinal chemistry to be able to design new types of molecules like antisense oligonucleotides or small molecules that can diffuse through the blood-brain barrier to get at neurons. We need sensitive biomarkers that tell us which patient subtypes belong with one another and which ones have a completely different type of disease process ongoing. We need study outcome measures that are highly sensitive to change that give us the first inclinations that a drug is being effective. We need those to be implemented at skilled clinical trial centers with research coordinators, research assistants, and principal investigators who know the disease well and how to assess it. And then, like the self-driving cars, we need artificial intelligence and data. So I thought that today, during ALS Awareness Month, in fact, the 30th annual ALS Awareness Month officially, first beginning in 1992, it would be a good way to begin by taking stock of where we are on these parallel developments that are bringing us to the precipice of the Holy Grail, which is truly effective, if not curative therapies for amyotrophic lateral sclerosis. So of course, ALS was first described in the literature all the way back in the 1800s, but the first ALS Awareness Month was announced and voted on by Congress in 1992. At that point in the United States, you were lucky if people knew about Lou Gehrig and his having and then dying of ALS. Maybe some people knew about Stephen Hawking and his struggles with motor neuron disease, but really there was not a lot of ALS awareness. Of course, there was amongst patients, people suffering with the disease and their families, and the clinicians battling hard, but not in the general population. A lot has changed over those last 30 years. We've had an influx of movies and biopics following people with the disease who have been honest and forthright with the American public about what it looks like to have ALS. Then, of course, we had the ALS Ice Bucket Challenge in 2014, which got a lot of people wet and brought in a lot of dollars, but I think almost as importantly, increased awareness amongst the American population, but also amongst our legislatures. And now I find that when I'm talking to people on a plane, when I'm running into them on the street, they've heard of ALS. They have basic awareness of what it is and understand the major challenge that it represents for people suffering with the disease and their families. Back when the first ALS Awareness Month began, there were essentially kind of three organizations here in the United States that were working on advocacy, clinical research, and the provision of clinical care. And there's been a dramatic increase, in particular since the ALS Ice Bucket Challenge. And shown here are just the logos of a few of the organizations that are battling this disease alongside of you and alongside of those of us at ALS centers. The dramatic increase in people working on ALS has dramatically increased the amount of funding that is available. Just to briefly review the current investments coming from major funders of ALS research, the ALS Association has raised and distributed more than $114 million for ALS research and care since 2014. The Department of Defense has invested a similar amount since 2009. Muscular Dystrophy Association, the NIH with $1 billion since 2009 have all played important roles in battling the disease. And I'm happy to announce that that investment continues. So the ALS Association just announced a new grant mechanism called seed grants to help get very innovative small projects off the ground to see where they go. The Department of Defense has actually doubled the budget for the ALS related research to $40 million in the next fiscal year. The MDA's Capture and Mover programs continue to conduct natural history studies of ALS patients. And many of you are involved in the NIH and National Institute for Neurological Disease and Stroke and their new strategic planning to figure out how to best implement ALS research. And then last Christmas, we all got a gift from Congress, thanks to the tireless legislative advocacy of people with ALS and their caregivers and the academic community and organizations like the ALS Association. The Act for ALS was passed, promising some $100 million a year for the next 5 years, which if you go back up to the previous line, is essentially doubling the NIH's budget over the course of the next 5 years. These important research dollars will go into expanded access programs for patients who do not meet inclusion criteria for standard clinical trials to get access to promising drugs, as well as to research grants to develop those next and important new round of drugs. There have also been important therapeutic advances, not only in ones that are approved by the FDA, but also in new clinical trial drugs. So back in 1992, so 30 years ago, the Riluzole studies were just wrapping up. And now in 2002, there were more than 100 companies at the recent Target ALS meeting, companies interested in developing drugs for ALS. Of those, 75 already have at least one drug in development programs for ALS. Shown here are more than 100 clinical studies that appear on clinicaltrials.gov and the different phases that they represent, which is a dramatic increase from just 30 years ago. In fact, when I was just starting to see ALS patients in 2009, 2007, 2009, there were 5 in clinical trials. And of course, now that number is getting close to the triple digits. There are so many, in fact, that organizations have developed clinical trial searches that can help link patients to clinical trials they might be eligible for. Shown here are the ones from the NEALS organization, from Antidote, and from IMALS, all of which are relatively straightforward to find if you are interested in participating in a clinical trial, ones that might be near your home, and ones for which the inclusion criteria might match your specific clinical circumstance. As I mentioned, it's not just the drugs that are already in clinical trials. It is, like I said, around 100 companies that are interested in developing ALS drugs, and more than 75 currently in development. One of the things that I find most promising about these drugs is that almost across the board, these are designed to target specific pathways inside of neurons or astrocytes or microglia that we know don't function well in ALS. From basic science, we've identified many of these pathways. I'll talk about a few of those in just a moment. But it used to be that you would find something that you thought might help neurons in general and bring that forward in ALS. Now, many of these drugs are targeting specific pathways that we know are dysfunctional. And I think from that, because of how they were developed, have a higher likelihood of being successful. I'm also happy to report that many of these companies are seeking very early access to ALS-specific expertise to help them think, even from the beginning of an idea, how it might play out across the entire development process for the drug. Many of them come to the Northeast ALS Association's Scientific Advisory Board. I sit on this and attend a meeting almost every other week where an early stage company will come, tell us about the pathway that they're targeting, give us their preliminary data, and then seek our feedback about whether this seems viable for ALS and how to go about developing it further. The FDA, with funding from the ALS Association, has developed a drug guidance document that helps industry understand how best to develop these drugs and the criteria being utilized. And then I think very encouraging is the fact that there are now several patient and caregiver advisory boards that companies come to, so that patients and caregivers are involved on the ground floor, providing input about how clinical trials should be designed, what inclusion and exclusion criteria should look like, which I think overall leads to far more successful clinical trials. Things that's coming along for Therapeutic Advances are large collaborative efforts to improve the efficiency of clinical trials. Shown here are 4 of the largest ones in the country. There's the NEALS organization covering North America, several countries in Central and South America as well, with more than 130 centers. There are 2 in Europe covering slightly different territories, one called NCALS and one called TRICALS, which serve very similar functions. And since the mid-2000s, PACTALS, which stands for the Pan-Asian Clinical Trials for ALS, and these cover Asia, India, Oceania, and have a large number of ALS centers as well. And these are designed to get ALS clinicians and clinical trialists working together, connected to patients, to be able to run more effective and faster clinical trials. A lot of smart people at all of these centers have been thinking long and hard about how to make the clinical trials that we run more accessible and more efficient. Many of you will have heard of the Healy ALS platform trial, the first of these new and innovative clinical trial designs. I'm not going to talk much about these, but there's a question and answer session later today about it. But the Healy ALS platform trial was the first to launch, comparing multiple drugs across very similar platforms. Just in March, TRICALS from Europe launched its first platform trial called the Magnet platform. And then in Scotland, a smaller platform trial called MND SMART has already been up and running for a number of years as well. And we fully anticipate that these new types of clinical trial designs will bring about, those 75 drugs that are in clinical development, bring them faster and quicker to clinical trial. We've also been working quite hard to reduce the burden of clinical trials. We know that as people get weaker and encounter more troubles in their activities of daily living, getting to an ALS center for a clinical trial is a major pain. And so we're all thinking about simplifying outcome measures and sorting out how we can incorporate both home and video visit assessments when possible to make it possible for people to remain in their homes even while good, reliable data is being accumulated. We also feel like it's important to increase access. Proximity to an academic ALS center has long been almost a requirement for participating in clinical trials because of the transportation time between your home and a clinical trial site. But we're working on increasing the diversity and the inclusivity so that patient populations and ancestral groups that have historically been ignored or overlooked can be involved. This also includes individuals who live in rural areas or further afield than possible for traveling to a center. And also, we're looking at ways to reduce the inclusion criteria barrier whenever possible to allow a broader swath of the patient population to participate. That when I first came to ALS research back in the 2000s, It felt very much like every academic center was working largely on its own with some overlapping efforts. Now it feels completely different. Now it feels like we're a large collaboration, different groups working, almost like an amoeba, like, moving and going together, pulling apart for the best research possible. And I think that that is well expressed in this next slide when I talk about advances in collaboration. So back in 1994, this paper here, when Rosen et al. Identified the first gene SOD1, the paper that came out had 33 authors representing 11 different institutions and covering 4 countries. There were 0 consortia involved in making that happen. It was mostly individual investigators in those different countries banding together. By the time we did our first whole exome sequencing project, something Debbie referred to in the introduction, we were up to 72 authors. So the number of people that were necessary to make these discoveries and working together had essentially doubled. There were now some emerging consortia oriented around genetics. And we now spanned 31 institutions across 4 countries. And a paper that I'll talk about in just a moment, just published before the change into 2022, which was successful in identifying 15 different ALS risk loci, again, doubled the number of people involved, now has 4 genetic associated consortia involved, more than 100, almost 150 institutions, and now spanning 22 countries, which I hope if you chart kind of the trajectory of collaboration here, you'll be encouraged that like never before, ALS researchers, patients with ALS, the centers, the institutions are working together to make the most of the research dollars that we have access to. When I first did my very, very first ALS Genetics Project back in 2007, I wanted to look at whether the spinal muscular atrophy gene SMN1 and SMN2 predisposed individuals to developing ALS. So I called up individuals across the country and asked if I could get some DNA from the patients that they had carefully amassed over the course of years. Only one said yes at the time. And in order to get that DNA, I had to send someone to that center. They went through the freezer and pipetted out, drop by drop, the DNA from around 500 patients, taking only the amount that we needed because it was such a precious and valuable commodity. Nowadays, things are completely different. We sequence basically everything all at one time, either by the whole exome or the whole genome. And one of the great advances in ALS collaboration has been the fact that we're all committed to sharing our data almost as quickly as it has been created and as quickly as it has been quality controlled to make sure that it's reliable. So now, if you want to know what mutations are present in ALS patients, you can actually go to multiple different organizations that are all posted online. So here are just 5 of them. They kind of appeared in the order in which they came online. But combined, there are now more than 20,000 ALS exomes or genomes available for searching, even from the comfort of your own living room, if you had the interest. And even more exciting is the fact that all of these are now being pooled and brought together into another effort called ANVIL, which will allow the genomes and exomes from all of these patients to be aggregated and then to be utilized by qualified researchers to come up with the next round of ALS discoveries. So I find this diagram particularly encouraging as we hit the 30-year mark of ALS awareness. So this shows the ALS research publications by year that are available on PubMed, the database that scientists use to find the scientific literature. And I think if you squint at this, you'll be able to appreciate kind of 3 phases of the pace of discovery. So the first of these is kind of a very slow, gradual accumulation of information while people were still relatively siloed and the field was just getting going. You'll notice that from 1992, the ALS Awareness Month beginning, a dramatic uptick in the number of ALS publications coming out each year. And then even more dramatically over the course of the last 15 years, has been really a skyrocketing of the amount of information that we have in the scientific literature about ALS. This represents the hard work of countless of scientists. And one of the things that I like to mention about the ALS Ice Bucket Challenge is that it didn't just raise a lot of money to do ALS research. It also brought in a bunch of people who have never thought about ALS, scientists who had never thought about applying their artificial intelligence platforms to a disease. People who had expertise in machine learning, people who have expertise in microglial biology, things that hadn't historically been associated with ALS have now entered our field and are helping. And that, I think, is one of the reasons why there's been such a dramatic increase in the amount of knowledge and the amount of publications related to ALS. One of the great successes and accounting for many of these increases in ALS papers are the discoveries of genes that are associated with ALS. Shown on this next slide is a timeline of the discovery of ALS genes. And actually, there are two additional ones that aren't on this yet because I hadn't had time to add them. So literally almost every 3 months or so, a new ALS gene comes into the literature, which is extremely exciting because as we discover each of these genes over the course of the last 30 years, we're learning more and more about what's going wrong inside of ALS neurons. When we take these genes that we've discovered, for example, and line them up underneath what the gene does for a given cell, you start to see some very important patterns emerge. You can see that a process called RNA metabolism, the generation of RNA, the degradation of RNA, the way the RNA gets shipped around the cell becomes an important theme in what's going wrong in ALS. We learn about the problem of protein homeostasis from looking at the neurons themselves under the microscope in ALS, but we also see that confirmed and reinforced by the genetics, where all of these genes here play a role in protein homeostasis, protein folding, misfolding, and recycling. As we look at these genes, we start to see a pattern emerge telling us that axonal transport and the role of the highway that goes down the very long axon plays an important role in ALS. And even tells us that getting the endosomes, little vesicles, little blobs and bubbles that have to travel up and down the axon, going in the right direction and to the right location is important. And these are important. Understanding these pathways are important because as we understand the pathways, they give us targets to go after for repair, for ways that we might be able to intervene in ALS in order to turn back the damage that's occurring and to help neurons, astrocytes, microglia, other cell types that are involved in ALS better function. So I just want to take a brief aside and talk about two of the exciting discovery papers in ALS genetics from the last year. The first one I already alluded to, which is the largest genome-wide association study ever performed for ALS, with nearly 100,000 cases and controls combined to identify parts of the genome that are increasing risk for ALS. So shown below here in this diagram is something called a Manhattan plot. Since I live in Manhattan, it's because now we have these super tall skyscrapers with $1 billion residential apartments at the very top. And so these are like the big tall skyscrapers, and the higher each of these skyscrapers, the more solid the evidence that that region of the genome plays a role in ALS. And prior to this study, we knew about 7. And so essentially this study doubled the number of regions of the genome that we know are associated with ALS. Now, in contrast to the gene mutations that many patients get tested when they have genetic testing in their ALS clinic, these are not genes that are super rare, where there's 1 abnormality per 100 or 2 abnormalities per 100. These, for the most part, are relatively common locations in the genome, meaning that if we can figure out what, for example, SCFD1, this little skyscraper right here, is doing to contribute to ALS disease risk, and can figure out a drug to either counter it or augment a process to counter it, we might stand a chance of slowing down ALS or preventing it altogether. And I think the promise of these types of studies is that with 8 new regions to go after, we now might be able to generate 8 new hypotheses, 8 new pathways that we didn't know were involved in ALS before that could give us important therapeutic targets. This is the second paper that came out in this last year. And I'm not going to talk about the details of this one very long, but I do just want to show that this group grew human cells in a dish. They modeled what was going wrong in those cells. They used computer learning. They took genetic data from humans. They took genetic data from mice, from brains that had been looked at with autopsy. They took really specialized genetic sequencing technologies applied to these organs, the brain, and the organisms, the mice, and then figured out clever ways to compile them all together to be able to identify that one gene, and this is just the first of the many that will come out of these types of analyses, the KANK gene, K-A-N-K, increases the risk for ALS by some relatively interesting mechanisms that remain to be studied further. But I just wanted to emphasize that the field of multi-omics, OMIC, which is transcriptomics, genomics, methylomics, basically all the different ways that we study cells can now be integrated, pulled together to give us insights into what's happening for ALS. And I do want to just share some relatively new data. We've now had the opportunity to look at 10,000 genomes from people with ALS. These have come from important collaborators. And as I mentioned before, people are sharing their data like never before. So involved in this particular study was the Precision Medicine ALS program that I run, the Biogen ALS Sequencing Consortium, a study that was entirely funded by the ALS Association called GTAC that I ran here at Columbia until last year, and then the New York Genome Center, which was mentioned in the beginning introduction, an organization that has received lots of funding from the ALS Association. Combined, we were able to put together 5,600 ALS cases and compare those to 14,000 controls. By looking at all of the genes that have ever been reported to cause ALS, we came up with the following plot. We found 451 of those 5,000 individuals carry the C9orf71 gene mutation. We found 73 that carry mutations in the SOD1 gene, almost 30 in the TDP43 gene, and then rarer causes of ALS with increasingly smaller numbers. All told, almost 12% of the people we sequenced had a mutation in an ALS gene. So we found some surprises. We found that some individuals who have been previously diagnosed with ALS actually carry causative mutations in hereditary neuropathy genes, genes that normally also cause sensory loss, but in some cases just cause motor nerve loss. We found mutations in the ataxin-2 gene, which usually causes spinocerebellar ataxia, but in some individuals can look just like ALS. The same was true for the Huntington's gene. Huntington's is a movement disorder that usually presents with abnormal movements called chorea, but we found Huntington expansions in our ALS cohort. Also, we found VRK1 spinal muscular atrophy and a handful of individuals who actually had Kennedy's disease as opposed to ALS. I think all of this emphasizes the importance of genetic testing in ALS, not just in people who have an obvious family history, but in everyone. And not necessarily just sequencing ALS genes, but sequencing the entire genome eventually, once the costs have come down, so that we're better able to detect these surprise genetic explanations that are becoming increasingly recognized in ALS populations. Now, I do want to just point out that the likelihood of genetic testing finding an explanation for a patient's ALS highly varies based on whether there's a family history of the disease or not. So we took 1,200 of the individuals who were sequenced, ones for whom we had reliable family history, and we classified them using a classification method from all the way back in 2012. Definite family history, individuals had 2 or more first or second degree relatives, like parents, or cousins, aunts, and uncles. For probable ALS, there was only one other affected individual. For possible ALS, there was another affected individual, but they were kind of far off in the family. And then individuals with no family history. And what I'd like to point out here is that in these two comparative studies, if you had more than 2 individuals in your family with ALS, we found an explanation on average 70% of the time by doing genetic testing. If there was only one other affected individual in the family, but it was your parent or your child, we found an explanation in about 58% of the time. If that affected individual was a second-degree relative, a cousin or an aunt and uncle or a sibling, and there was no one else in the family that was affected, then the likelihood of finding an explanation drops to 30%. If there's a rumor of someone in your family who had ALS or somebody who is not a very close relative, the number is relatively similar to if you have no family history, right around the 10 to 12% range. And I'll just point out that even in people who have been told they have sporadic disease, we do still on occasion, 1 in 100, for example, find a causative gene mutation in individuals who didn't know there was a possibility of family history. Because other data is readily available, I told you about the Project Mine data browser from Europe that was available. I was essentially able to go immediately to their database of 4,000 ALS patients, combine them with my almost 6,000 and get to the 10,000 number. And I won't show you any specific data from that because essentially it shows identical factors. We see the same frequency of ALS genes. We see the same number of people whose diseases are solved by genetic sequencing. I would also just point out that there's a lot of stuff that we didn't find when we did sequencing of 10,000 ALS genomes. In particular, we found a bunch of genes had 0 mutations. I showed you that big long chart with all of the gene mutations, and it turns out that some of those are extraordinarily rare. And we're now in the process of going back and reevaluating the genetic evidence for those genes to see which ones actually cause ALS and which ones don't. That's important going back to our diagram here. To basically cross some things off this list is almost as important as adding new genes to it, because it will help us ultimately refine which of these pathways we focus in on in order to develop more successful and more efficacious therapies for ALS. So I'm about to wrap up, so be thinking of those questions to put into the chat. But I hope that over the course of the last half hour or so, I've convinced you that we are at a break point for the development of really highly efficacious ALS therapies. Like developing that Tesla that can drive on its own, all of the different pieces had to progress to the point where they could work together to bring about self-driving vehicle. And I really feel that we're at that same juncture for ALS research. The clinical trial networks are in place. The medicinal chemistry and the large throughput screening technologies to figure out which ones of those help in ALS cell models and animal models are in place. We understand so much more about the disease than we ever have before, thanks to the massive number of scientists and the research dollars that are coming into the field. And it's my great hope, and I'm sure you share that as well, that in the next years, we will see the tipping point where we start to have our self-driving vehicle version in ALS. And with that, I'll go ahead and pause and stop sharing and open up the floor for questions.
Great. Thank you so much. I was feverishly taking notes because I was learning so much. So I want to pose a few questions to you that were submitted. The first is if you could just expand a little bit on the whole idea of biomarkers and how that is being developed and what your hopes are for that field.
Absolutely. So just briefly, a biomarker is something that we can measure in a living patient, either an MRI or looking at the spinal fluid or looking in the blood, something that we can look at while someone is alive. that gives us additional information than just the clinical examination. So it could tell us, a biomarker could tell us whether someone was at risk to develop disease. It could tell us whether somebody would progress faster than average or slower than average. And biomarkers are very useful for lots of different reasons. If you're looking at people who carry gene mutations but don't have symptoms, a biomarker might tell you who might go on to develop symptoms in the short term. It might also tell you who won't go on to develop symptoms, which would be also very helpful. A biomarker might also allow us to find a group of patients who are predicted by that biomarker to progress more quickly. And so we could take that into consideration when we do our clinical trials to make sure that people at risk for fast progression are evenly distributed between the placebo group and the control group. And that would make clinical trials smaller, and it would potentially make them shorter. So there are a lot of different reasons to develop biomarkers. And actually, one of the things that I didn't talk about today is some exciting news on that front. Researchers have identified that the gene called TDP43, or the protein that comes from the gene, is essential for regulating a cellular process called splicing. And when TDP43 isn't around and doing its job, which is often the case in ALS, some abnormal splicing products are visible in the spinal fluid and sometimes in the blood. And so for the first time, over the course of the last year, we're learning that we might be able to use those to tell who has TDP43 still working pretty well, who has TDP43 that's not working well at all, and to be able to use that as a biomarker. So thank you for asking about biomarkers. That's one particularly interesting and hopeful thing that's come about in the last year in biomarker development.
Great. And then we have a question here. Somebody asked, she says, I have ALS and a parent with ALS. So where in the New York City area can I get my genome sequenced?
So in most cases, we wouldn't start by sequencing the entire genome. That still is a $1,000 prospect. And we would first begin by doing clinical sequencing. Clinical sequencing is done in something called a CLIA certified laboratory, meaning from the time the DNA arrives to the time that it gets looked at, there are all sorts of safeguards to make sure that the right answer is achieved every time. So the first step in identifying a gene, particularly in a family, is to have clinical genetic sequencing done for known ALS genes. At the moment, that happens to be free via a number of mechanisms. So there is a sponsored testing program at Invitae Genetics. There is a sponsored testing program at Prevention Genetics, more or less looking at a similar number of genes and both covering all of the important ones. And if your clinician isn't familiar with those, they're more than welcome to email me, and I can have either my genetic counselor or myself let them know how to go about getting that genetic sequencing. One thing that I didn't mention, and I should just pause here because I would be derelict not to, is that when you sequence 28, 30 ALS-associated genes in someone, 25% of the time, you'll find something that you don't know how to interpret at this time. These are called variants of uncertain significance. And when you get a variant of uncertain significance, that's exactly what it means. We don't know what it means. And a lot of people get very worried about these variants of uncertain significance. And if that were to happen in your genetic sequencing, I would strongly recommend talking to your clinician about it, asking what's known about those particular mutations. And then the organization that I'm helping to run called the ClinGen ALS GCEP is highly involved in trying to sort those out, trying to figure out which variants actually play a role in ALS and which ones are just benign, because we suspect that most variants of uncertain significance don't actually play a role in ALS. They just happen to be differences between different humans. So lots of complicated things to discuss, best handled with your clinician.
Thank you very much. We have another question. And what the gentleman asks is, he says, you mentioned the importance of genetic testing. Yes. Is there any evidence indicating the likelihood of getting ALS if an individual's genetic profile shows a C9orf?
Yeah, so let me just restate that. So if you have someone with no symptoms of ALS who's carrying a gene mutation that we associate with ALS, there are kind of two questions to ask. One is, what is the statistical likelihood of going on to develop ALS or a related symptom during the course of life? So people have tried to estimate this for the C9orf72 gene for really any of the genes, SOD1, TDP43. And because of how rare these are and how small families are and how short a period of time we've known about the C9 gene in particular, we don't have a nailed down answer. And it probably differs from family to family based on what other environmental, lifestyle, and other genetic factors might be present. But I will say encouragingly that the longer we study C9, the less likely we think it is that people will develop ALS. We used to, from the initial studies, say that there was virtually 100% chance. If you lived to the age of 80, there was a 98% chance of developing either ALS or frontotemporal dementia. As we've studied more families, as we've studied people with no family history who have C9, we're learning that that number is not that high, and it keeps coming down. We best guess it's probably around 50%. It could be lower than that, but it will take us a few years to be able to tell. So that's one way to look at it. Like just statistically, if you carry the gene mutation, what are the chances of going on to develop symptoms? But now getting back and kind of linking this up with the biomarker question from before, we're hoping that we will be able to find biomarkers that might predict which of the 50% or which of the 45% or whatever that number finally turns out to be are at the highest risk of developing ALS. Because if we develop a preventative therapy, you would love to employ that only in people who are at risk of going on to develop the disease. So you could not do spinal taps all the time for the person that you knew was not going to develop symptoms. And this brings me to one of the other things I didn't have time to talk about today, and that is the tremendous amount of research that's taking place in pre-symptomatic gene mutation carriers. So here at the ALS Center at Columbia, we have the ALS Families Project, which was launched with funding from Project ALS and has been supported by other funding agencies since then, where we see patients carrying gene mutations back on an annual, sometimes biannual basis, depending on what people can swing, to look for those biomarkers, to basically get spinal fluid, get blood, do very careful clinical assessments so that we can discover those biomarkers that will predict who may or may not go on to develop disease. And I think that will be a linchpin to developing preventative therapies, in particular for those individuals who have gene mutations. While studying those people who carry gene mutations, though, we're learning things about all of ALS, because it's very likely the case that the biomarkers we discover that predict when someone with C9 mutation might go on to develop symptoms will have applicability to people who don't carry gene mutations. And so we're hopeful that, like in Alzheimer's disease, the things that we find in people with gene mutations immediately carry over and give us insight into people who don't carry ALS gene mutations.
Thank you. So then I think a good follow-up question to that is one that was asked about the role of genetic counseling and how that plays into this, if people are having, it's being sent to Invitae, what does a person do with this information? How are they guided?
Yeah.
How is that handled?
Yeah, so it depends on the clinic. And this is actually one of the major challenges kind of facing the field of genetics across the board. There are far too few genetic counselors in the world. Training programs are ramping up to try to meet the demand, but we need for medical reimbursement strategies in the United States to catch up. As it is right now, we have to raise funds from philanthropy and other mechanisms to pay for the genetic counselor that works at our center. So at the ALS Center, patients have access to our genetic counselor because essentially it's being subsidized by philanthropy because it's not a billable medical procedure. So Ellie Harrington, many of you have probably met her, spends up to 2 hours sometimes with patients explaining some of these complexities. And that time is completely unrecognized and unreimbursed by the US healthcare system. And that means that if you don't have philanthropy to cover a salary like hers, then you're not able to provide often the type of genetic counseling that I think is appropriate and should be provided with genetic testing. I will say this, the organization that is funding the genetic testing at Prevention Genetics has gone the extra mile to fund genetic counseling at Prevention Genetics. So they have contracted with genetic counselors to be able to provide some of that. So that may be one route forward, particularly for centers or for people who don't have access to genetic counseling. Now I'll just back up and say, genetic counseling doesn't have to be done by a genetic counselor. Neurologists and ALS experts like doctors, nurse practitioners who know their way around this material can provide competent genetic counseling and then call in their help from their colleagues, the genetic counselors, since there are so many fewer of them when complicated situations are encountered. And so there are a lot of strategies for tackling that, but I completely agree that genetic counseling should be provided. And the next time people are marching on Capitol Hill, that would be a great thing to tell Medicare so that genetic counseling could be provided as a clinical service at more centers.
Okay, duly noted. Just a couple more questions. One person is asking, she says, my mom, she or he, I'm sorry, says, my mom has dementia and I have developed ALS. Could there be a relationship between the two?
So the first clues to a link between ALS and dementia, in particular frontotemporal dementia, as opposed to something like Alzheimer's disease, was first suggested at UCSF by a very good colleague of mine who helped train me when I was a resident, named Kathy Lomenhirth and Bruce Miller. And they noticed that about 15% of people in the ALS Center had trouble with executive function, and went on to kind of study those individuals in better detail and found that 5% actually developed frontotemporal dementia, like it was diagnosable, met all of the criteria for that. And maybe as many as 50% of ALS patients had some slight deficits that you could find on detailed testing in that domain. And that link really started to bring our two fields together, the dementia fields and the ALS fields. And that link was fostered by the discovery in 2006 that when you look in the neurons of people who have passed away from ALS, you find abnormal TDP-43 protein. And when you look in the brains of people who die with frontotemporal dementia, you find abnormal TDP-43 protein. And this suggested that some very similar mechanisms might be causing a problem. Since then, we've learned that many ALS genes can cause frontotemporal dementia. Many frontotemporal dementia genes can on occasion cause ALS. The major culprit in those is the C9orf72 gene, which explains the majority of people's families where there's both frontotemporal dementia and ALS. There are others, but that's an initial thought. Now, if it's Alzheimer's disease, that's very common. And so it's not uncommon for us to see patients who have a loved one with Parkinson's disease or Alzheimer's disease or some other neurodegenerative syndrome and wonder about a link. And there are some loose links there, but it's not nearly as strong as if the type of dementia is clearly frontotemporal dementia. So now when I talk to patients, I do ask about a family history of dementia, and I think that does increase the likelihood of finding a gene mutation, in particular, the C9orf72 gene.
So the last question goes back to something that you touched upon early in your presentation. It's a little off the specific topics of genetics here, but it has to do with clinical trial design. And if you could just say a few words about how the industry, how the field is attempting to create innovative new trial designs that could help either broaden the inclusion criteria, which you referred to, or at least make it a little more accessible, I guess, to a wider variety of people that would like to participate.
Yeah, absolutely. So the first thing I'll say is that I'm in the lab sequencing genomes and trying to interpret genomes more than I am conducting clinical trials. I do some, but you'll definitely want to direct some of those questions to Sabrina Paganoni and the panel that's happening later today about clinical trials and research. I guess from my perspective, what I would say is, I'm not leading any of those efforts, but I do see my colleagues putting on their thinking caps and talking with a really broad swath of medical researchers across the board. So, platform trials, for example, which I spent some time talking about, come directly from oncology, where you take one group of tumors, one type of tumors, and people get randomized to several different therapies in parallel. That allows for fewer placebo patients, for example. It allows for an ongoing trial infrastructure that basically lasts for years without having to kind of recreate a new clinical trial operation every time a new drug comes around. So I think that's one way. There are other types of trial designs that are called adaptive trial designs, where you stop at intermittent time points and decide kind of which direction you should go. And if you've pre-specified when those time points are and what the decision tree is, you can maybe make a trial shorter or need to extend a trial if it's looking promising, but you need a little more data and you think the drug is working. So there are lots of people, not me, thinking about how to make that happen, and you'd get a much more robust answer from Sabrina and others later in the session.
Okay, and two more questions have slipped in, so
Yep,
I'm gonna put them out there to you. The first is, what is the impact of COVID on ALS research? And the second, I'll just put it out there and you could figure out how you want to frame these different, is do you have any information about stem cells and ALS?
Yeah, absolutely. So I'll start and just say that COVID has been a double-edged sword. Obviously, with the first wave coming through and most academic medical centers shutting down, the number of delays for clinical trials and so forth was enormous, and has definitely set the field back in some cases by a year or more in terms of getting clinical trials that were supposed to happen off the ground and completed. I would say that like at Columbia, where the administration recognized that ALS is a terrible disease and that these clinical trials were super important, ALS research suffered less of a brunt than many other fields with kind of more chronic or more common diseases where people just didn't feel the research was worth exposing people to the risk of COVID to bring them in. So we did get more done in ALS clinical research than many other fields did during the major wave of COVID. But I think, that's the downside. The upside is that we all had to figure out how to use Zoom and had to think very quickly about how to get clinical trial grade information from people while they're in their homes. And I think that has, ushered in, the field of wearable devices to collect data, of doing as much virtually as possible to save people from having to come in. And so there's been a tremendous upside to things that we've learned during the pandemic that are being put into place in clinical trials quickly, I think. And then stem cells, is a very tricky thing to talk about. So I'll pretend that each of you is sitting in my clinic room and has asked me this question, and I'll say that I firmly believe that stem cells used to nurse sick neurons and to keep them alive for longer will one day be part of how we treat ALS. The idea is that you can take stem cells and either engineer them or coax them into producing what I call happy juice. So growth factors that neurons like, anti-inflammatory molecules that might shut down microglial inflammation, molecules that might help axons stay alive for longer. And you'd like those floating in the spinal fluid, kind of bathing the neurons that are under attack from ALS. And I do think that we'll get there one day. There are several challenges, and that is that you need to engineer those stem cells so that they produce enough of that happy juice. You need a lot of growth factors. You need a lot of anti-inflammatory molecules to have the kind of effect we're looking for. The other thing that they have to do is they have to do it for a long time. And I think the current generation of stem cells dies off pretty quickly when it gets infused into the spinal canal. And they don't hang around long enough producing the happy juice to have a strong impact on the cells. Some of the clinical trials that we've seen so far, kind of provide a little hint that they might be helping in certain subgroups of patients to a small degree. But I think what that does is tell us this is a fruitful direction to head, but we need to get cells that live longer and secrete more of those factors that will help keep the neurons alive before it's really viable as a tool in our armamentarium. So stay tuned, because I think it will happen. We'll get there. We're just not there yet with the current generation of stem cell therapies that are available.
Wonderful. Dr. Harms, thank you so much. Your presentation today, in addition to being really, really informative, was very inspiring. It's clearly an exciting moment to be in our space. And I too have been in this space for many years, and it's thrilling. It's really thrilling to hear all the work that you and your colleagues are doing. And I'm sure that our audience shares that sense of inspiration. So thank you again.
Thank you for having me.
Absolutely. And Dr. Harms already gave a plug for Dr. Paganoni's presentation later on today. Our next presentation, our next session is going to begin at 1:30. It's called Understanding Frontal Temporal Dementia, which Dr. Harms referred to with an FTD ALS diagnosis. So we hope that you'll stay tuned and come back in for that. Again, Dr. Harms, thank you so much, and have a nice afternoon, everybody.